Yesterday, what was the purpose of what we did yesterday? Joe, electrophoresis. What was the purpose of that process? Arch. To do what?
To separate the
mixtures.
The mixtures based on
Based on what?
Charge and size.
Charge and size, right? All right. So chromatography. This is another separation technique, right? And you guys, I want you, as we're doing it, I want you to think about what's similar and different about these two techniques. All right. So chromatography, basically, there are two phases, right? Phases in this sense doesn't mean like a time period, right? Usually phase means like some sort of like time period. Phase in this case means like substances, right? So there's one called the stationary phase and one called the mobile phase. What do you think about those? What can you tell about those? George?
One of them, it doesn't move, and the other one does.
Good. Which one moves and which one doesn't move?
Probably the stationary one doesn't move, and the mobile one does move.
Good. So it's basically like there's this, you put something through this medium that's not moving, and the thing that's moving is the one that's going to help separate the thing that you're trying to separate. All right? So some types, and you guys might have seen this, and you might have even done it in middle school. A lot of people have done paper chromatography before. You put like a little dot of ink on a piece of like special paper, and then you put like liquid, and then the liquid helps carry the ink to the other end of the paper, and it separates them in the process, right? So originally the ink like started out as black or green or something, and then as it travels through, so in this case, what's the stationary phase then? The paper, right? And then this like liquid would be the thing that's helping move the ink or like that marker or whatever through this paper, right? And there are other types like paper, liquid, gas, column, etc., right? But today we're going to work on column chromatography, okay? Good? Any questions so far? Okay. So basically, so what is column chromatography? So we're starting with this thing. This is called our column, right? And we're going to fill it with this thing called matrix, right? And matrix is made of a polysaccharide, and that's why we're doing it in this unit, because we just finished doing what? Carbohydrates, right? And polysaccharide is a type of carbohydrate. So it's made of this solid matrix, and that's known as the stationary phase. So this matrix isn't going anywhere. It's just staying there, and something is going to be passed through it to be separated, right? And then so what we do first is we take this matrix, and you'll have a little beaker of it, and you're going to pour it into your column, right? And then you take this liquid called elution buffer, and you just add it to the top with a transfer pipette. And this process, if you pour this matrix in, and then you pour a water, like a liquid thing on top of it, you're pretty much like making it sort of condense down, and that's called packing the bed. Kind of cute, right? So you're going to pack the bed first when you get your columns and when you get your matrix, right? So again, we said the matrix is a stationary phase, right? If you notice down here, do you want the matrix to just like fall out into your cup here? No, that would defeat the purpose. So it needs to be supported. So it's supported by this thing on the bottom, and here it's called a porous plug, but it's also called the frit, F-R-I-T, and that's going to support the matrix. But it allows water or liquid to go through, but it holds the actual matrix up. Because which one has like bigger molecules, do you think?
Carbohydrates.
Yeah, the matrix will have those. So this is kind of like a filter thing that lets liquid go through, but then it supports the matrix, so the matrix isn't going to fall through. All right. Then, so we have our thing. We have our bed all set. Then what we're going to do is we're going to take our mixture that we want to separate, and we're going to put it on the top. So that's this black thing that we're putting on the top, right? And as it passes through this stationary phase, it's going to start separating, right? Is it just going to go down here by itself? It doesn't. It will go a little bit, but it doesn't really want to go that much. So we have to move it down there by applying this thing called a solvent, or by adding. The solvent is more elution buffer, basically, right? So we're adding more elution buffer from here. It's the same thing. We put more on top, and it's going to start pushing the dye, or like the mixture, down, right? If it's pushing it by moving it, what phase do you think the elution buffer is? That's called that's the mobile phase, right? So that's moving. This is the mobile phase, and then it's also called the eluent. And these words get really kind of crazy. So the eluent is the elution buffer, and it's pushing the sample through the stationary phase. We're good? Okay. So do you see that it's starting to separate here? Here's the green and here's the red. And as you add more elution buffer and it starts to elute, so the verb elute means to pass through and then separate, right? So it's eluting through this stationary phase and it starts to do this. You see what's happening there, right? It starts to separate. And then what you guys are going to do is at the bottom, you're going to start collecting the separated parts in fractions of 0.5 milliliters in these little tubes. Okay? So I'm going to apply the sample, and then it's going to pass through the column, and you guys are going to collect them in 8 little tubes. Are we good? Yeah? Okay. So that's the process that we're going to do right now. Are you guys ready? All right. These molecules, you're going to collect them in different ones. So some of your tubes might be red, or like some of your tubes might be one color, and then the color might change over the 8 tubes that you're collecting, all right? But you have to make sure that you're ready to quickly, once you get one tube, make sure another one's ready right there to collect. So you want to collect them continuously. You want to collect 8 samples of 0.5 once you start seeing the colors coming down. All right?
Does it drip out slowly, or?
It's pretty slowly. It's not like gushing out,
Okay.
right? Yeah. Okay. So you don't have to yeah. But make sure you're on top of it and make sure you're doing good teamwork dynamics, right?
So is color the reason that they separate, like, or
So-
do they just turn different colors?
No. So let me just shh. All right. Let me just tell you sort of what's going on here, right? What just happened? Did anything just happen there? No. And the stuff that's coming out here is called the LU8, right? Which is like the stuff that you're collecting. You're collecting the LU8, right? Okay. Are we okay? You can always like refer back to this later. So basically the matrix, if you zoom into the matrix is going to be also, it's if you look at it, it's composed of all these little beads. Do you see these beads here? These big things are the beads. They're, right? You see them?
Yeah.
Yeah? Okay. So, and presumably the size of the molecules that we're separating, like the colors, not just the colors, but it's that they are composed of different sized molecules, right? So these beads have these little pores inside of them, right? Which molecules can fit inside of those pores? The smaller ones, right? So the smaller ones will actually go into the pores in the beads and kind of play around in there, like get knocked back and forth and like get, sort of dilly-dally along. And then, so what will the speed of the green ones be versus the red ones, or the small ones versus the large ones?
The large ones will move fast.
The large ones will move faster because they can't even fit into these beads, right? Because the pores are too small. So they're just going to be like clunking on by, right? And so I like the analogy of like, if you picture all these animals like storming through this like kind of row of houses, right? So there are all these houses here, right? There are like bunnies, let's say. There are like deer, and then there are elephants, right? So the elephants, can they fit into the houses? No, so they're not going to get distracted by the houses. They're just going to stampede on through, right? But the deer, can they go into the houses? Yes, they can, but can they go into the rooms? Let's say they can't, right? So they go into the houses, so they're playing around a little bit, but then they just go right through, right? But the bunnies, what can they do? They can go into the houses and then into the room. So they're getting delayed, like really delayed. So the largest ones elute the fastest, the medium ones then elute later, and then the smallest ones elute last because they're kind of like getting knocked around and sort of getting stuck inside the beads themselves, right? Does that make sense? Right? So what factors are we separating these based on? We're separating them based on their size, right? Think about the shape too. What's so funny? Are you laughing at me? Oh, okay. All right. So let's suppose I have rod-shaped molecules here, right? Like rods are like kind of like long things, right? Can the rod things all necessarily, like if a rod is facing this way, is that going to fit into that pore? No, right? So the rod's going to just do what? Just going to go right down, right? Whereas like a sphere, can the spherical things fit in the? Yeah, they heck yeah, right? So this is actually shape. So the factors, when you're asked what factors does this technique separate things based on, it's based on the size of the molecule, the weight of the molecule, which is kind of similar to size, but then also the charge. I'm sorry, not the charge, the shape, right? Size, weight, shape. So the longer rod-shaped things are not going to fit in the pores, so they're going to elute faster also. Faster than spherical ones, right? Okay. Are you guys all set? Okay. So I still have a few more things. I want to talk about two different types. Sorry, I want to talk about two different types of chromatography after we do this. I want to make sure we can do this, right? But I just have to show you one more thing. So once we collect our little samples, right? We're going to have 8 little tubes of our samples, right? Some are going to be one color and some are going to be another color, right? If I look at color, what kind of observation is that? Qualitative or quantitative? Qualitative, right? But then if I put them in this machine called a spectrophotometer, I can actually measure the wavelengths of light absorbance, right? So if something, sorry, I'm going to do this. If something appears yellow, right? Can you guys find yellow on the color wheel?
No.
Right? If you're colorblind, sorry. All right. If you find yellow on the color wheel, a substance that's yellow means that it's doing what to yellow light?
It's absorbing.
It means it's absorbing everything except yellow, and it's reflecting yellow back to your eyes, right? So what will something that appears yellow, what will it absorb best?
Purple.
Good. And what wavelength is around purple? That's around, let's say average, that would be like 420, right? All right, and what about something that's blue? Something that's blue, that appears blue, what's it gonna absorb the best? Right? And what is it? Yeah, we're actually gonna set it at 620, I think. But so it's gonna you recognize that something that appears a certain color actually absorbs the opposite wavelength the best. You can get that, right?
Yeah.
So that's what we're gonna do. You guys are gonna put your little samples in a spectrophotometer, right? That's gonna actually measure not only, like, you can visually see it with colors if you have color vision, but if you don't, you can also put it in the spectrophotometer and trust that it's gonna tell you, based on these absorption patterns, what color, like what it's absorbing the best, right? And that means you would know that it's yellow if the thing that is yellow is the one that absorbs 420 light the best. Okay? All right. So I want you guys to come over here. I want you each to get a handout because I want you to do these as we're going, but I want you to put on your gear, and then I want everyone to get a ring stand at their table over there. Right? I want 5 tables, and everyone should have a ring stand. And this is, obviously you have to finish this for homework, right? Okay, so, all right. So, all right. Just leave your little column at your table. That's fine. Just leave the column. Try to put your ring stands back. Try to put beakers and racks and other stuff back. All right. So, what were the 2 factors that? What were the 2 factors or the 3 factors that we were using to separate these molecules from? Ro?
Size, weight, and shape.
Size, weight, and shape. Right. So, which ones eluted the fastest? The small, spherical, light ones, right? And then which ones elude? Oh, sorry. Just kidding. The fastest ones were what? The large ones. The large, heavy. I'm sorry. I was thinking of the other thing, and then I said my voice just took over my body. All right. So yeah, the largest ones, the rod-shaped ones, the long ones, and then the heavy ones elute fastest, and then the other ones, the small, spherical ones, elute the slowest, right? All right. So that's one way. That's one type of column chromatography, right? But if we want to change the matrix, if we change the nature of the matrix and we change the nature of the beads in the matrix, then we can also separate different things, right? So, for example, there's one called ion exchange, which basically, instead of making matrices with little holes in them, beads with little pores in them, I make normal beads, but then I have charges on the outside of the beads, right? In this case, what charge are the beads? Positive, right? So if I put a mixture of positive and negative things, like a mixture that's mixed, and I want to separate the positive and negative things, which ones are going to stick to the beads? The negative ones. So which ones can I separate from each other? So I can get the positive ones to elute first, and then eventually the negative ones will kind of let go and come out also, right? But that's one way that I can change the nature of the beads in the matrix to kind of change my objective. So that's separating them by charge, right? So that's ion exchange. And then the last type is called affinity. So in this case, I make specific beads, right, with maybe a specific outer portion that will attract another complementary another thing, right? So do you see how these red ones have that little rod, that little like stick-shaped thing in them? Right? So if the beads are shaped like that, like complementarily, then they will stick to those things, right? And then the other ones which don't have that shape are just going to elute faster, right? So that's called affinity, and then that's based on like the outer like sort of nature, or that like the outer, like the specific like molecular interactions of the things that you're trying to separate, right? So you see how you can like change the nature of the matrix to kind of like get your objective of separating the things, right? Arsh?
A question about like gel filtration. So like the rod ones, those would be quicker, right? Because they don't have to go to the
Because they can't get into the pores, so they won't dilly-dally. Yeah. Right?
So would that like separate by like a surface?
So it separates, yeah, basically by the nature of the surface, right? And I have a question. The more something interacts with the matrix, the faster or the slower it's going to elute?
Slower.
The slower, because it's like playing around, right? And so it's like, you know, hanging out in the matrix, and then it won't come out faster, right? This is interesting, isn't it?
Yeah.
It's crazy that you can just like, you know, be like, okay, I want to separate these things. Oh, I'll pour it through this thing, and it will sort of like filter them out, right? It's like kind of blind faith that you're like doing, you know, like, oh, okay, I guess those molecules are smaller than the other ones. But it's really cool that someone figured this out. I mean, can you imagine like figuring this out? Okay. So again, why do we want to separate mixtures?
Just see how they compare to others.
Yeah, well, we could just see what something's made of, first of all. We could like purify them, or we could analyze them. So there are a lot of different ways that we might want to do this, right? And then why are we doing this during the carbohydrates unit or after the carbohydrates unit? Sorry, George.
Because the matrix was like the one that was breaking it down. So
Yeah, the matrix is a polysaccharide itself. So is the gel and the agarose, right? The agarose gel. But also the blue dye is also a polysaccharide, which is kind of interesting, right? So you guys have that packet. Read the packet. Like if you don't understand what you did at all today, that's probably a problem, because you're going to be quizzed on. So on the lipids quiz, it's going to contain questions on this, questions on gel electrophoresis, and questions on lipids, which we're starting tomorrow, right?
no quiz this week.
I'm considering. Well, would you rather have it on Friday or Monday?
Monday.
I think that we need the time. So I think that what I'm going to do is we're going to do Monday. So tomorrow and Thursday we're going to introduce the two lipids units that I want to do. Friday we're going to start. You guys are going to work on the lipid research project, and then Monday you'll present the lipid research project and take the quiz on Monday. Is that okay?
So is the quiz shorter?
The quiz is short. It's a pretty short quiz, yeah. Oh, actually, I'm not sure because I have to put these two labs in it, so it might be a.
Well, maybe I'll be done by 1.
I think you'll have to come during lunch no matter what. So, but whether you want to come that day or the next day is totally up to you. All right.
I agree.
Where's Arnav's cell?
Is Miss Wilson here today?
Bro.